In this video, we will discuss about the mechanisms that occurs in the Invadopodia and how the E-cadherin is related to that. So, in the heading, from the heading it is clear that it's the E-cadherin ARP3 complex that helps in the Invadopodia formations in the pancreatic cancer cells. So, how they come up to this point? So, in order to find out the mechanisms behind these E-cadherin roles in the Invadopodia formations, what it is, they did the full mass spectrometry analysis of the conditions where E-cadherin is depleted and compare it with the conditions where E-cadherin is present. And when they did the analysis, they are able to see some of the proteins that are upregulated and downregulated. And when they use those upregulated and downregulated proteins, they are able to find out the pathways that are dysregulated. And these are the list of the pathways in the panel A that are mainly dysregulated. In this, their focus is on these actin nucleations by ARP-WASP complex. The reason why they focus on this is that in the previous literatures they are able to see the role of ARP in the Invadopodia formations. So this is the main reasons, one of the main reasons why they focus on this ARP-WASP complex. And when they check the genes that are involved in this ARP pathways, when this E-cadherin is downregulated compared to the control case, they are able to see that the genes that are involved in this ARP pathways are downregulated compared to the case where this control silencing RNAs are used. This further substantiate their finding that this ARP3 might be interacting with the cadherins, or ARP3 plays a role towards this Invadopodia formation in the pancreatic cancer cells. And they check this ARP3 actin formations, and thereafter they check this ARP3 actin formations and also did the Western blot to check this ARP3 amount in the case where this E-cadherin is knocked down, and they are able to see that this ARP3 complex formations is less in the case where E-cadherin has been knocked down. And similarly in the Western blot also they are able to see a decrease in the ARP3 concentrations compared to the case where E-cadherins are present. So this is the top panel is just the ratio of this Western blot where ARP3 by actin shows. And after they also did the immunostaining or PLA experiments where they checked the ARP3 and cotinine complex are present, and simultaneously they also check this E-cadherin and ARP3 complex are also present. Then this indicates that this ARP3 cotinines and E-cadherins makes a complex that helps in the Invadopodia formations. And in order to validate their complex formation hypothesis, what they did is that they prepare cell lines where ARP3, they prepare cell systems where ARP3 is being silenced or knocked down, and then check the Invadopodia formations. So here the validations of this ARP3 silencing is shown in the panel F, where we can see that amount of ARP3 formations goes down significantly compared to the control case, and the Western blot of it is also shown here. And then they check the number of Invadopodia per cells, and they are able to see a decrease in the Invadopodia per cells in the case where ARP3 has been knocked down. So this confirmed their hypothesis that it's the E-cadherin ARP3 complex formation that helps in the Invadopodia, and if either of them are absent, then they will not be able to there is a decrease in the Invadopodia formations in the pancreatic cancer cells. So this is how they validate their points that E-cadherin ARP3 complex helps in the Invadopodia formations. And this panel A is the results of the mix of the proteins that are upregulated and downregulated, and the list that is here shown are of the pathways that are dysregulated. So this could be either upregulated or downregulated. The separate list of the upregulated and downregulated not shown in the main figure. So I hope you understood this, how they validate this hypothesis and how they come up to this ARP3 complex. That is from the mass spectrometry analysis of E-cadherin knockdown cells and E-cadherin's positive cells. If you want to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.